You're watching a Science Bridge video about the streaking technique. What is streaking? Well, streaking is a technique for finding a single trait from a bacteria population. In the transformation lab, we will be using two populations of bacteria. One population of bacteria will have the DNA plasmid and will be shown as a positive sign. The other population of bacteria will not have the DNA plasmid and will be shown as a negative sign. These bacteria will be grown on an agar plate. Two of these plates have a red tag. Plates with the red tag contain AMP or ampicillin, and will be explained more in the later part of the video. Step 1. Add 100 microliters of the positive bacteria from the microfuge tube onto the positive plate. If you're not sure of your pipetting skills, this would be a great time to review them. Step 2. Add 100 microliters of the negative bacteria from the microfuge tube onto the two negative plates. Now that you have added the bacteria onto the agar plate, it is time to spread them around. When you spread the bacteria around, make sure to do so evenly. When you first add bacteria onto the plates, do not wait long for streaking, because the bacteria will clump. Also, make sure that you're gently spreading the bacteria on the agar plates. Imagine that you're spreading whipped cream on top of Jell-O. I know, doesn't that sound delicious? Finally, put the plates upside down to grow overnight in a 37 degrees incubator. Now, why did we just use 3 plates and not just 1? And what does the LB and AMP or ampicillin do? LB or lysogeny broth is the food for the bacteria. AMP or ampicillin is an antibiotic and will kill or slow down the cell's growth. And of course, no ampicillin means that there is no ampicillin in the plate. The reason we had 3 plates is because 2 of them will be the controls and the last will be the experimental. The LB no AMP minus only contains food. If the bacteria were not able to just grow on food, that may mean that the bacteria may have been cooked during the heat shock, or that there are no bacteria to begin with. This plate is a control for bacterial viability or bacteria growth. LB AMP negative contains food and ampicillin. Because AMP kills bacteria, if any bacteria grow on this plate, that means that the AMP or ampicillin is not working. This plate is a control to test if ampicillin is working. LB AMP positive has food and ampicillin. Because these bacteria have the plasmid, they are able to resist the ampicillin and thus grow. But what in the plasmid allows the bacteria to resist ampicillin? Well, the plasmid is a circular piece of DNA. The plasmid has multiple genes, and one of the genes is resistance for ampicillin. Because of this plasmid, the bacteria is able to grow on the ampicillin plates. Other proteins the plasmid can code for can be the fluorescent proteins. Because the ampicillin gene and the fluorescent protein gene are on the same plasmid, we can easily grow the bacteria we want by using this technique. Congratulations! You have learned the streaking technique!
Yay!